Hello, everybody. It's really exciting to be here and show you guys what I've been doing for the past few years. So part of this project that we call the PlanktonScope, which, as you'll see, is involving like a lot of very great people and a lot of people doing a lot of cool parts of this project. And so what I hope to show you by the end of this talk is how, opening up these projects can really bring things to new heights and approach problems that might not otherwise be possible. So we'll jump right into it. So I think, again, with this problem, which we've opened up to a huge community, we've been actually able to approach these large problems. So anybody studying the ocean knows like the ocean is very big. So studying the ocean is a huge problem, especially when you're looking at microscopic things. And so what we're interested in is plankton. So these are any microscopic organism that's kind of at the mercy of the ocean's currents to kind of float around. And while they're small, they're extremely important, right? They form the base of a lot of these global food webs, and they sequester, through making their bodies anywhere from 40 to 60% of the carbon dioxide emissions that we're putting out on any one day. And if you're wondering why that figure is kind of so vague, it's because we just don't have enough information on these plankton assemblages to ground truth a lot of these models. And if you see that little red dot I have there, that's actually like in this image, like 10,000 times too big if we're actually going to represent the amount of the ocean that we've actually explored. So when we were looking at really small things in these droplets of water, this is like a huge problem. And so we need to approach this in a way, where we could scale these instruments in a very massive way to look at this very massive sample. And so you might wonder how plankton can actually help us. So why are we interested in imaging plankton? And the thing is, plankton are really amazing kind of integrative biomarkers that can provide us a really good readout for ocean health. So because they have very quick generational times, we can actually get really rapid information on what's going on in the ocean at any one part of the ocean. So for instance, based on these metrics of say composition, diversity, and just total abundance, we can get readouts on a lot of these problems that are happening. So for instance, with ocean acidification, a lot of these calcified planktons will not do so well. They won't be able to form their shells, and you'll see a real difference in the diversity indice and like in the plankton composition. Microplastics we can directly see as readouts in our imaging system, as well as, with pathogens, with these harmful algal blooms, you'll see a definite drop in the diversity in these various regions, and you'll see these blooms very clearly in these plankton images. And like what we can do with this is, not only through research where we can, understand more. So a lot of researchers want to know why this stuff is happening, how it's happening, and how we can help prevent it. But we can actually get, again, we can ground truth these ocean models, so actually know more on how climate change is affecting our oceans through these plankton assemblages, and kind of start making more accurate conservation efforts and ecosystem management. So actually, on the direct level, when we have these algal blooms, we could actually, start closing beaches or, inform people that there is toxic algae in the water, and maybe they should be aware of that and, not consume stuff that's coming out of the water. And so one of the giant barriers to research is not just this massive sample that we're looking at, so this enormous body of water that is the oceans of our planet, but how expensive it is to actually research this. So there's a lot of, very amazing people and a lot of very amazing instruments that have, for the past couple hundred years looked at plankton. We've known they've been important. But these research vessels are extremely expensive to run, so they're anywhere from like $40,000 to $80,000 a day just to run the vessel. And in this, they're extremely complicated and they can't do many deployments. And their reach is, very small. So the sample size is very small when we're trying to understand the ocean as a whole. And again, there are some very nice and amazing instruments that do exist for looking at plankton, but they're also very expensive. So which puts them out of a reach of a lot of people that really need to look at the plankton. So these people that are actually really dependent on the oceans for living. So these instruments are also very complicated, which makes it kind of hard. There's also often a lot of downtime with these instruments waiting for repair or service. And often people are kind of tentative to open up these machines because they are so expensive and complicated. And so the Planktoscope project has a lot of objectives, but we really wanted to democratize access. So through this kind of open source maker community, make accessing plankton images much lower cost, which we can then empower a lot more people to get involved. So we can get plankton imaging in the hands of more people, educators, citizens, researchers, and these underserved communities, which are often the ones that are most impacted by, especially these harmful algal blooms, and they often lack the facilities and money to afford the research at such a high level. And from this, we can generate massive data sets, right, that are beyond the reach of a few single instruments and a few research vessels. And through crowdsourcing, right, we can get, a diverse amount of people looking at the data and kind of creating like a massive data set that we can all use to understand the ocean in a better way. And at the base of this is just taking good images, right? What we want to do is make an instrument that is, democratized, low cost, but also still taking data that is intercalibrated and of very high quality. So research quality images that we can use as a group, as a community. And if you are aware of Manu Prakash, so that's the lab I'm in at Stanford University, he's shown through many different diagnostics, but showed how through frugality, so making instruments more affordable and accessible, we can approach these problems of scale. So if we can make instruments that are very high quality, but at much lower cost while maintaining the performance, we can approach these large problems and put science in the hands of people that might not be able to afford it otherwise. And so in this way, we've seen a lot of great success in actually getting, high quality instruments at lower cost, but into the hands of people that might not be able to afford it otherwise. And so how we've done this, we started, right before COVID, and it was kind of a hard time for anybody that was trying to do a community-driven project. But we've seen that there are an incredibly diverse group of people that are very interested in the ocean. So whether they are makers, scientists, citizen scientists, or just people that like to build things with their hands, we've seen that there was an incredible amount of interest for people that wanted to replicate this instrument and kind of add data to the vast amount of data that we wanted to collect on the plankton communities. And so through COVID, we had over 100 people making these microscopes. So we put the plans up online, we put a few guides on how to replicate, and it was honestly just really humbling and very impressive to see how resourceful people are making the instrument out of an incredibly diverse body of materials and kind of hacking it, making it in their own way. But we saw people, were able to replicate this, and we're super excited to take data on plankton and really help us make the instrument better. So through this community, actually, we've iterated many different versions of this device, and it is where it's at now because of this community, using the machine, making the machine, hacking the machine, and making it much better. And so what is the actual PlanktonScope? So this is the version we're at now. Again, this went through a lot of iterations, and that was honestly made possible by this community using it. So I have a background in, protein biochemistry. So I was used to using, again, very expensive, kind of inaccessible machines that I didn't really understand. And so when Thibaut Polina joined the lab, who got me involved in this project, he kind of came from this maker community and showed that these things don't have to be so complicated. So we kind of broke down what a flow-through imager needed to be and what we needed to do to get high-quality images. So what we needed was a pump. We needed, obviously, a camera to take the images and some lenses in which to look at these images, and then a way to focus. And this was all done through, I'm sure we're all aware of the Raspberry Pi, but it's an incredibly powerful, but affordable computer to kind of do the processing on the machine. And through this, we kind of broke the machine down to parts. And in that way, it wasn't as intimidating anymore. So what we did is have a lot of consumer-based parts. And so this would come in a kit, and this comes in a kit now that you can get. So you break it down, and it's really not as intimidating, right? You have this pump, you have your Raspberry Pi, you have a way to drive the pump, and a way to store the data. And we came to the shell of the machine, or the body of the machine, which can be made, in any of these maker spaces that have a CNC machine. And we focused on trying to make it out of the most sustainable yet reliable and durable material. And we made it out of this, right now, this bamboo, which is actually bamboo flooring, which is really incredible. It's great to machine. as well as being really durable and actually waterproof. And so if you come to this with kind of like the mindset of putting together like an IKEA desk chair or something, in a few hours, you can take all these components and put together your PlanktonScope. So within a few hours, with even not a lot of experience in building, you can start taking images with your PlanktonScope. And this is kind of how I like to see the PlanktonScope, because really it's at home, just throwing it in your backpack and taking it out, and you can run this off just a little battery cell. And so you can take this anywhere you want in the field, and look at samples and start taking data with nothing more than the PlanktonScope and your cell phone. So the software architecture was also made to be very open. So it's built on this Node-RED platform, which is this kind of flow-based programming software, which is also easily hackable and modifiable. So the nice thing with the PlanktonScope is with the Raspberry Pi, it generates its own Wi-Fi, right? So all you need is a laptop or a cell phone with no installed software. Everything is on the actual PlanktonScope itself. So the PlanktonScope will generate its Wi-Fi, and you can just connect through your cell phone or computer. And in that, you're able to just connect to the machine, and then everything is on the machine. And through that, right, you can control, so you can load your sample either directly, or you can take your plankton tow or whatever you want to look at, and start actually running image acquisition. So you can start taking tens of thousands of images just from your cell phone. And then on the Raspberry Pi, after you take these images, you can run these segmentation algorithms. So what you get is your raw images, which will all be on the Pi. And then you can run segmentation, which will then find any objects that are in those images. So in this way, it's really nice, right? You're not really reliant on concentrating your sample, because even if there's not much in the images, the segmentation will pull out anything of interest and then store that as an object on the machine. And this way, it's nice because, say you're just interested in these objects, you can extract those objects, those plankton objects, with the metadata and store those, and then ditch the very large raw images. Say if you're on a sailboat or something and you don't want to have to store, gigabytes of data. And then when you do get back to land or wherever you have network connectivity, you can then upload these to what we're using now is this beautiful system called the Ecotaxa database, which is where you can upload your plankton images, and there's actually hundreds of very qualified marine biologists which can then validate your images and tell you what's in there. So you can get species level identification of those objects that you found in your image acquisition. And so this is just showing, the kind of simple segmentation process that we use, where you have your raw images. It'll then subtract the background, then use kind of a simple thresholding, and then pull out your actual regions of interest. And so in the current setup that we have, which is using conjugated M12 lenses, which are these very high quality but frugal lenses, you can get down to, like 20 micron resolution up to anywhere to 300 micron resolution. And this is, again, all changeable by changing the flow cell or by changing these lenses. If you're interested in something much larger or much smaller, those are all easily modifiable with our kind of modular setup that we have. And so what you got, so these are just from two different imaging projects, one that was in kind of brackish water, and then one that was in water off Croatia, as we put this on a sailboat, a few months ago. And so you get all these giant data sets filled with all your different objects, so all your different plankton objects. And then you can do with this what you want, right? So you can then upload this to Ecotaxa and then, get those species levels confirmations from these experts. Or, you can do whatever you want with these images, in addition to making these like very beautiful collages that you can look at, which get, a really good view of what is in this water that you're interested in. And so again, like I said before, flow-through imaging is not new. There's a lot of these instruments that do this. One of the most well-known is this FlowCam. And so what we did is TiVo imaged the same sample through the FlowCam and the PlanktonScope. What we saw is, with our machine, which is, orders of magnitude less expensive, we're able to get, equally, if, not in my opinion, better images of these plankton with the PlanktonScope. And so you can get these research quality images on this machine that, we've built ourselves. And so again, what do you get? You get these, hundreds of thousands of objects of plankton. And again, you get these beautiful collages, which I really love looking at. But like, what do we want to do with these objects? So what can we do with these plankton images? And so I'll go through a few of the uses that we have, starting with, research. So I'll start kind of selfishly with a plankton that I'm very interested in, which is this pyrocystis. And so there's another problem with plankton in that over time, right, plankton really changed the way they look. So pyrocystis especially goes through many different changes in its life cycle. And so it's really nice to know for me, because I was really interested in how this organism affects, carbon sequestration, and it does this through this interesting life cycle and the way it migrates through the water. But these plankton, the same species even looks very different depending on where it is in its life cycle. And so with the PlanktonScope, I was able to take, tens of thousands of images and make these different classes. So I was able to take these segmented images and then see that this one species of plankton actually looks very different over the course of a few days as it goes through its division cycle. And if you're kind of naive, you would say, my goodness, these aren't the same plankton species, but in fact they are. So with these tens of thousands of images and a lot of these other really beautiful open source resources we have, which make these, at this point you can make extremely good object classifiers with just even now a few hundred objects, and then go back to your plankton images. And over a few hours using these beautiful open source object detectors and TensorFlow, I was able to take those segmented images and then go back to the raw plankton images. And then in my culture, I'm now able to make these very good classifiers and detectors. And over time, with very high confidence, say exactly where my culture is in the different life cycles. And for this, I was able to see a lot of very cool things on the diurnal capabilities of this thing and how it actually lives in the ocean. And so again, these are all things you can do in a few hours, right? You can make these very high quality object classifiers and detectors, whatever you're looking for. And you can imagine, say, if you are interested in some harmful algal bloom that was happening, you could easily train one of these on that algae of interest, and then, be able to detect this with very high confidence in your sample if that's what you wanted to do. And going back to algal blooms, if you're not familiar, these are a huge problem that are only becoming more massive of a problem. And so these are when an algae that either directly, so through creating a toxin, which a lot of these algae do, or by actually just sucking out all the oxygen from the water, can be a huge problem, right? So in an economic sense, they can really devastate aquaculture. So a lot of these communities are really dependent on shellfish farming or fish farming. And so with this, they're changing the water by adding, a lot of fertilizer or food. And in that, you can create these blooms of harmful algae, which will actually take over and kill the shellfish or the fish that you're looking at. And in many communities, so we did a lot of work in Indonesia, they can be very harmful to humans. So if you consume shellfish that have accumulated this one dinoflagellate, you can get this paralytic shellfish poisoning, which is really devastating health-wise. And it's very strange because it's very hard. The symptoms can be hard to diagnose initially, and the actual toxin is very almost undetectable unless you have very advanced imaging to look at the actual molecule. But one thing that is very easy to see is the actual algae. So using microscopy, we can look at this algae, detect when it's becoming very highly abundant, and then make this kind of a sentinel or an early warning device to look at the algae bloom. And this is something well within the means of doing a flow-through imager. Again, this is a problem. If you look at the satellite image, you can see like it changes very much depending on where you are. So a lot of these surveillance initiatives we have are just limited by the personnel to sample the water as well as the instrumentation to actually look at the water. So if we can get more instruments out there looking at, geographically more spots in this water, and kind of enable the community to do their own, monitoring, we can, do this in a much better way and let people know where these blooms are happening to, warn people well ahead of when they're going to get sick from these algae accumulating in the shellfish or fish. And so again, we did this a few months ago with a beautiful initiative out of the University of Maine and a few others. And so we went to Indonesia, where this is becoming a huge problem because the water quality is really alarmingly changing over the past 15 years, where this paralytic shellfish poisoning on a lot of islands is becoming a huge problem. And a lot of these people depend on fish not only for their livelihood, but also they're sustained on fish for their protein. And so what we're able to do is take in one transect. Take the PlanktonScope on one of these boats and train a lot of these fishermen that are actually out there. and have them taking images and uploading these images, in a few days. So in this, we can see, you can enable people that are actually on the water to take charge of these projects and form their own initiatives looking for these things which really devastate the communities. And going back to aquaculture, again, so aquaculture is also really dependent on knowing if you're farming mussels or oysters, you really need to know when these things are spawning. So in order to accurately farm these, what happens is you put these dishes in the water and these larvae will land on the dishes, and then you'll get the start of the farming of these oysters or mussels. But in order to do that, you really need to know when these larvae are spawning in the water, because if you put these dishes in too early, they become biofouled, and then the larvae will not want to land, and you'll basically completely lose, or almost completely lose your harvest of these oysters or shellfish. And so with the PlanktonScope, what T was able to do is go and put this in the water and then see very accurately the different stages of these larvae. And so in this, if you were sampling either automatically or just going out and sampling once in a while these oysters are going to be spawning, you can get a very good look at exactly when the larval stage is happening, and then you could much more accurately put in this infrastructure to then get the oysters accurately landing and then get a good harvest. And then again, for free, right, as you're imaging this water, you could also see if something bad was happening, right? So if you had a bloom of Alexandrium or something that was going to cause this harmful algal bloom, you'd be able to see that in your data and say, again, you could very easily train an object detector to tell you, okay, we have all these different larval stages, which is very nice, but we also have this bloom of Alexandrium. So maybe we should think about, when we should put these plates in, or, maybe put these somewhere else. So in this way, like through this plankton assemblage, we get a lot of information on the water quality, because, this plankton assemblages really accurately encode a wide range of signals, both environmental and human influence that are in the water. And so where we want to go with this is, bring more autonomy to the plankton monitoring. So right now, we do have these things where it's very nice. So you can put these plankton scopes really anywhere you want. But what we want is to have these, very autonomous. So the vision is, right, to have, a buoy with a plankton scope on it. So autonomously, it's going to be able to take images and, even say, again, if you found a harmful algal bloom, even just send you a text message saying, look out, we have these algae that are blooming right now, so you should be aware of this. And that's kind of the vision that we're going at. Every day we're trying to make this better. And honestly, it's been, again, really great through the community. And so what we'd like to do is just complete and build more of a community so we can get this co-design and include more people to make this better. And again, this has been actually really great. We've got a lot of great ideas for how to build autonomy into our plankton monitoring through this community. And so again, this is really great. So we really hope we can get more people involved in the PlanktonScope project. And I really encourage you to go to the website or the Slack. And this is a great way that you can kind of interact. There's hundreds of people on this community already. And Thibaut and I are on there. And so we really love to hear what people want to see out of the PlanktonScope and really how we can make this better. And in this way, we can kind of make it better and kind of get more ambitious with what we want to do with this plankton data. And so we've already have like 1 million images that have been uploaded to Ecotaxa, and we hope to, kind of exponentially increase this number as we go through, so we can be more ambitious and kind of look at more plankton and approach more problems with the PlanktonScope. And yeah, so I just want to thank, again, this has been a huge project, and I can't really thank everybody that's been involved, but it's been really amazing. But the true heroes are Thibaut and David. So Thibaut got me involved in the whole DIY community and got me involved in plankton, actually. So it's been like a wonderful journey. And Dov has been really heroically making a lot of these plankton scopes and making them better and kind of getting these designs to the point where they're extremely robust now and able to be put together in a few hours and taking plankton wherever you want. And of course, Manu Prakash, who is very inspirational in showing that, yeah, with frugal science, we can still do very good science that involves a lot of people that might not have access to it otherwise. And of course, Stanford and Fairscope, which is the entity that kind of came out of the need for making more plankton scopes in an intercalibrated way. So you can go to Fairscope and get help getting a plankton scope and get help with actually putting together one if you want to. Again, thanks again for everybody for coming. This was really fun, and I'd be happy to take questions you guys have about PlanktonsCope or plankton in general.